So in yesterday's video call, we discussed Bryophyta, Pteridophyta. For those of you who are on it, we discussed Bryophyta, Pteridophyta, and gymnosperms. Now what I'm going to do is I'm going to do a quick recap of, not an in-depth recap, but just a quick recap of certain concepts that we encountered when we dealt with Bryophyta, Pteridophyta, and gymnosperms. Gymnosperms obviously being the first section of your spermatophyte. Now, when we looked at bryophytes, one of the main things that we saw here, we saw that the gametophyte generation was the dominant generation. We saw that the gametophyte generation was the dominant generation, and the sporophytic generation was the temporary generation. One is permanent, one is temporary. We saw various things here, but one of the main things we saw when we were doing bryophyta, we looked at them. So I'm just gonna do a quick recap on what happens here with bryophyta. So what happens is, when we are doing bryophyta, remember the dominant generation is the gametophyte generation. So the gametophyte generation will start first. Now in the gametophyte generation with bryophyta, we see that gametes are produced. Now these gametes, obviously we know that we have male gametes. So gametes are coming from the male sex organ, right? We have a male sex organ and we have a female sex organ. Now a male sex organ and our female sex organ have names. The male sex organ is known as the antheridia. Antheridia sounds very male, sounds like Andrew. Alright, it's known as the antheridia. And the female sex organ is known as the archegonia or archegonium, right? Now what happens here is, obviously we know, remember we are still in the gametophyte generation, we know that the antheridia is going to give us sperm and the archegonia is going to give us our ovum / ova. You can use either one there, right? What we know here is what is going to happen now is, we know that between the sperm and the ova, the process of fertilization is going to take place. And in this process of fertilization, the sperm here, it needs water in order for fertilization to take place with the ova or the ovum. Once fertilization has taken place, we form a new structure now known, and this new structure is now known as the zygote after fertilization has taken place. And this zygote will now represent the sporophyte generation. And now what happens is, that zygote will develop structures, right? Now if you have the drawing in front of you, good, because then you will know what structures I'm referring to. The zygote will develop something known as a foot. The zygote will develop something known as a foot, and that foot anchors the sporophyte generation to the gametophyte generation. And then it's also going to develop something known as a seta. And the seta is what bears the sporangium. The seta is there to bear the sporangium. And then we also see, it obviously the third structure here would be the sporangium. Right? Now what happens is, the sporangium, if I'm just going to draw the zygote part, I'm not going to draw the gametophyte part, but when we're looking at the zygote part, what happens is here, we see that, okay, this is your gametified generation, you know. You have your female branch here, you have your male branch, and you have your rhizoids there at the bottom. But out of it develops a foot and a seta. At the top of it here, we see that there is sporangium. And on that sporangium, we see there's something known as a calyptra, which is like a cap on top of it. Right? So we can see here that the foot with the spot here is anchoring the sporophyte, this is the sporophyte generation, to the bottom part, the gametophyte generation. And then out of the foot, we see this long structure develop, which is known as the seta. And on the seta, we can see that the seta bears this part here, which is known as the sporangium. And the sporangium, at the top of the sporangium, we see the calyptra. Oh, what was that? The calyptra. Now what happens is, inside the sporangium, there are spores. Right? So inside that sporangium, we will see spore formation. So inside the sporangium, spores will develop. Remember, this is not inside here. Inside the sporangium, spores will develop. And what happens is that sporangium will dry out. The calyptra will fall off. And it will release those spores into the environment. Those spores will then be dispersed by wind to damp, shady environments, and then what we see is that the spore will then start the gametophyte generation again, and that is the alternation of generations. When the one generation ends, the other one starts, and then it ends, and then the other one starts, and then it ends, and that is the alternation of generations. It's a cycle, and that is what we looked at when we dealt with bryophytes. Now when we looked at pteridophytes, and obviously we know with bryophytes that there is no vascular tissue present, and it's our most simple part of the plant kingdom. But however, when we moved on to pteridophytes, we saw a change here. Remember, in bryophytes, the gametophyte generation was a dominant generation, the generation that you see all year round. However, here we see that the sporophyte generation is the dominant generation. The sporophyte generation is the dominant generation. That is the first thing we want to know here, that the sporophyte generation is the dominant generation. Now, if I'm just going to do the fertilization part. So we know how a fern leaf looks. Now, on the underside of the fern leaf, this is going to represent the start of the sporophyte generation. On the underside of a fern leaf, on the underside of the spur of a fern leaf, either in clusters like this, or singularly, we see sporangii. And when it is in clusters like this, we call it sori. But when it is individualistic like this, we call it sorus. Right? And this is the start of your sporophyte generation, which means it has to do with spores. Right? And now we know that. So what happens is the spores are released from the sporangia and they are dispersed by the wind. So these spores over here, all of these spores here are released. So these spores released. These spores are released into the environment. And they are then dispersed by the wind to shady, moist environments. So Now what happens is, once it reaches its environment, its optimum environment, right? What we see now is that this spore will germinate. And this will now germinate gametophyte. Remember that. So it will always, so when something germinates, it's going to the gametophyte generation. So these spores have now reached the soil, or wherever it is, damp soil, and now it is going to germinate. And this germination, once the spore germinates, it's now going to represent the start of the gametophyte generation. Now what happens here in the start of the gametophyte generation, we see that something known as a heart-shaped prothallus develops. A heart-shaped prothallus develops, and this heart-shaped prothallus has rhizoids here at the bottom. It has what we call, obviously, our male sex organs, or your antheridia, and then it also has our female. It has female sex organs known as the archegonia, and then it has an invagination. Right? It has an invagination. Now what happens here is the male sex organ will obviously give us sperm once again. So our male sex organ will give us sperm, and our female sex organ will give us ovum. Now these, the sperm and the ovum, needs to fertilize. But here with Pteridophyta, also the same with Bryophyta, we see here that this sperm needs water for fertilization. That sperm needs water to travel to the ova. So now it travels to the ova, and we see that fertilization now takes place. Now obviously we know when fertilization takes place, a structure is developed known as a zygote. And now we see our zygote developing. Right, and our zygote develops, or we can even, a zygote forms, or we can just call it a new fern in the sporophyte generation. We can also say it's called a young sporophyte. But our zygote develops, so this would now again then start off the sporophyte generation. So now we're going back to the sporophyte generation, and now our zygote develops. Now our zygote develops, or our young sporophyte. What happens is, so our zygote has now developed, representing the sporophyte generation again after we have now fertilized, and it brought about the end of the gametophyte generation. What we see here with this is that the prothallus is still there, right? The prothallus is still there. At the top of the prothallus, we will see our young fern plant. At the bottom, we see our rhizome. We see our the start of the adventitious root system that is developing for the young sporophyte, or the young fern plant. And then we also see that this prothallus will still have some rhizoids here. Right? Now what happens is that this young sporophyte, it lives parasitically off this prothallus over here. Remember, this is our prothallus here. This is seeing it from this view. You are seeing it now from this view. Remember, when I did it, I showed you from that view. Right? You saw the female sex organs, the male sex organs, and the rhizoid. I showed you like that. You would have seen the rhizoid, you would have seen the male sex organs, you would have seen the female sex organ, and you would have seen that invagination over there. But now it's in this type. It is in this view, horizontal view. We saw it upright. Right? So that prothallus, now that prothallus has developed, If the prothallus has developed and the young sporophyte is developing, but this young sporophyte is going to live parasitically off that prothallus. It's going to steal its water and mineral salts because remember, its adventitious root system is not fully developed for maximum uptake of water and mineral salts. Therefore, it's going to use this rhizomes because it's all one structure. And eventually, this young sporophyte will develop into a compound leaf, a massive fern plant, and it will no longer need the prothallus. This prothallus will then die off, and the rhizoids leaving, and the rhizome will stay there. And what we will then see is that when our rhizoid is horizontal, our compound leaf will grow out of it. Our rhizome. A compound leaf will grow out of it. Remember, this is your rhizome over here. Our compound leaf, a massive fern leaf, will now grow out of it, right? A massive fern leaf will now grow out of it, and then at the bottom here, we will now see our adventitious root system that is now fully developed, the adventitious root system, and now we can live independently without that prothallus. But the main thing that we want to highlight here is that young sporophyte lives parasitically off the prothallus, right? It lives parasitically off the prothallus. Eventually, the prothallus dies, and what happens is the young sporophyte will now become a compound leaf, and it is independent, so it can take it. The adventitious root system has developed, so now it can have maximum uptake of water and mineral salts. That was Pteridophyta. Then when we got to gymnosperms, which is the first part of the spermatophyte group, which is the first section of your spermatophytes. So your spermatophytes are divided into two sections: your gymnosperms as well as your angiosperms. But we are going to deal with the gymnosperms first. Now what happened? So the plant that we want to use, that we use when we're explaining gymnosperms, is a pine bear tree. Now with gymnosperms, first thing I want to make known here is that, remember when we were dealing with bryophyte and pteridophyte, we just spoke about the gametophyte generation, finish and clear. But now here we speak about something called the male gametophyte generation, as well as the female. I'm not going to write that, but if we speak of the male gametophyte generation and the female gametophyte generation, just keep that in mind so long. And the reason is because here the male spores go through their own process and the female spores go through their own process. So we can't refer to them collectively as the gametophyte generation because different things are happening to them. Remember with bryophyte and pteridophyte, the spores were undergoing the same process, but here male spores will develop into a pollen tube, and your female spores will now develop into an ovary with an ovum inside of it. And the other three will die off, but let's look into that. So we have a male cone and we have a female cone. So they look similar, but what happens on these cones are very different. So our male cone here will have pollen sacs on it. Come on, man. We'll have pollen sacs on it. Inside of these pollen sacs, we will see pollen grains. Inside of these pollen sacs, we see pollen grains, so this is your pollen sac. Then we see these dots here are pollen grains, or we call it male spores. Okay, I wrote some pollen grain and male spore because I'm referring to one only, I'm not referring to more. But they are pollen grains and male spores. Then when we're looking at our female cone, when we're looking at a female cone, remember this is a seed scale. This is known as a seed scale. These are seed scales, right? So on the seed scale, what happens here is we see the formation here of an ovule, right? We see the formation of an ovule. Let me just see if I'm using the correct term. Yes, we see the formation of an ovule. Now this ovule, inside of this ovule, we have 4 female spores. So this here is an ovule. Inside of it is our female spores. Don't say female pollen grains because it's not female pollen grains. It's female spores. Right? Now what happens is the male pine cone will release its male. The pollen sac will burst. It will release its pollen grains. Right? So if this is the pollen sac here with the male spores inside of it, and this here is our ovule with our female spores inside of it, what we see happening here, is we see that this pollen sac bursts and it releases those pollen grains or those male spores. Now what happens is, and it normally happens in spring, so what happens is these male spores will then travel to here. They will stop here. They will travel to the ovule, right? And once they have now made it to the ovule, right, they will now, so now they have made it to the ovule. Oh, that's a bit too big. Oh, look like Pac-Man. They will now travel. Oh, and before I go on, I just want to make note here that only one female spore survives. The other 3 die or disintegrate. Alright? So this one female spore now, it becomes the main stick now. That one female spore will now take up its space. Alright? And what happens here is that, okay, just throw it nicely. This male spore develops into a pollen tube. This male spore develops into a pollen tube. And this pollen tube, right, this pollen tube contains 2 male gametes. Contains 2 male gametes. Remember, this is now that spore, right? But it's not, we don't call it a spore anymore. We now call it the female gametophyte, right? And inside of this female gametophyte, we have our female sex organ, also which is known as our ocagonia. Inside of it. Oh yes, before I continue, each female spore, I forgot to say that, each female spore, remember there's 4. Now each female spore individually will have 2 to 3 inside of it. It will have 2 to 3 female sex organs, which is ocagonia. Right? So this is a female spore, or the female, you can also call it the female gametophyte or the male gametophyte, because this is known as the male gametophyte and that's known as the female gametophyte. Anyway, but inside, you just need to know now that this is that spore. This is the singular spore, it's now gotten bigger, and inside the spore there's a female sex organ, right? And then, so there's a female sex organ, and this is obviously the male sex organ. Now what happens is, we will see here that the one male gametophyte will fuse with that female sex organ and fertilization will then take place. So we will see that these 2 will fuse and we will see that fertilization will take place. fertilization will then take place, and we know when fertilization takes place, only one, right? Remember, only one survives here. We can't have two because it's not double fertilization like in angiosperms. Here, we have two male gametes, but only one male gamete survives. We have 4 female spores, but only one survives, right? So it will fuse, and then it will have fertilized, and then we will form some, and then again, here we form the zygote. After fertilization has taken place, we always form the zygote. So now we will form the zygote, and this zygote will then form an embryo, and that embryo will then form a naked seed. Not a covered seed because it's not double fertilization. It forms a naked seed. Now let me just go over it again. And let me identify where is the male sporophyte generation and where is the male and the female sporophyte generation. So the male sporophyte generation happens here. This would be the male sporophyte generation. The male sporophyte generation is the releasing of that pollen. Right? Then when this pollen tube forms here, that is the male gametophyte generation. Right? That is where the male sporophyte generation and the male gametophyte generation is. Then when the 3 disintegrate, right? That is here your female sporophyte generation. Then when only one spore survives here, that is known as your female gametophyte generation. Right? And then obviously here when the zygote forms, it will form an embryo, it will form a naked seed. Those naked seeds will then be dispersed by the wind, and they will end up somewhere in a favorable environment. It will grow a pine tree again, and then we will see male and female pine cones. So eventually here we will again see a male cone and a female cone. And then that will then represent again the male sporophyte generation and the female sporophyte generation. So that is your gymnosperms. Remember, gymnosperms is single fertilization. Then when we're looking at angiosperms. Now angiosperms, you can go and read the textbook. There's a lot of information about angiosperms. But because I didn't do it last night, I'm going to do it fully now. So the angiosperms are part of the sporophyte group, which we said. And we obviously know the sporophyte generation is the dominant generation. They're the most advanced plants, most successful for life on land, for terrestrial life, or the most successful terrestrial plants. They are non-thallus, which means they have 2 root stems and leaves. They are divided into 2 categories, which is known as your monocot or your monocotyledonous and your dicotyledonous. So angiosperms are divided into 2 sections, your monocots and your dicots. Right? Now obviously there are some differences between monocots and dicots. First difference here is that monocots, their flowering sections will always occur in multiples of 3, and the dicots will occur in multiples of 4 or 5. Right? Then monocots will always have an adventitious root system, whereas dicots will have a lateral root system, or we say a taproot system. And few other differences here, which we will see when we get into it, is that there are structures in a flower known as the corolla and the calyx. Right? There are structures in the flower known as the corolla and the calyx. With monocots, the monocots corolla and calyx, they are fused together. And when they are fused together, we call them the perigon. However, the perigon, right? And then here with the dicots, the corolla and the calyx, they are separate. They are separate. And the last difference here is in the venation. So monocots will have a parallel venation system or parallel veins, whereas dicots will have a net venation system or net veins. Make sure you know this table for if they ask you to tabulate 2, 4, 5, 2, 1, 2 of, they can ask you to tabulate 1 to 4 differences between monocots and dicots. So make sure you know this table. Obviously not like this, you need to write that forward. Florals occur in multiples of 3, florals occur in multiples of 4 or 5. It has an adventitious root system, it has a taproot system or lateral root system. The corolla and the calyx of monocots are fused together and is known as the perigon. The dicots, corolla and calyx are differentiated and therefore they are separate. They have a parallel venation system and they have a net venation system. The leaves have a net venation system and a parallel venation system. That is when we're talking about your monocots and your dicots. Now, when we're speaking about angiosperms in general, let's just quickly talk about certain sections of it. Now, obviously, we know that angiosperms is the most advanced, so angiosperms will have vascular tissue. Vascular tissue being your xylem. Xylem is able to obviously transport water and mineral salts, and then phloem is able to transport food. It will have xylem and it will have phloem. Then we also know it has a stem. Now this stem is upright, first of all, very rigid like an oak tree. It has strengthening tissue in order to keep this plant upright. So it's upright, it has strengthening tissue. Right, the stem also bears the leaves, and what is so, I'm not going to write the full sentence, but it bears leaves. If we look at a tree, for example, the stem bears leaves at the top, right, which makes it possible for maximum absorption of sunlight, so maximum photosynthesis, and then also it makes it it makes it, oh my English, the stem bears the leaves for maximum sun. Okay, there we go. The stem bears the leaf at the top or in areas where it can get maximum sunlight and carbon dioxide for photosynthesis. Maximum sunlight and carbon dioxide for photosynthesis. Then also we see here, it will position its flowers, because angiosperms are flowering plants. So it will position its flowers in places where a pollination can happen easily, either insect pollination, animal pollination, bird pollination, or wind pollination that can take place, right? Because not all plants can self-pollinate. We don't have to know pollination for this paper, but not all plants can self-pollinate. Most of the time we see cross-pollination happening. Like for example, a bee will sit on the stigma, and the stigma, it will then take that stigma to the anther and blah 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 blah, which we can look at now. And then we also see with angiosperms that its leaves, the main function of the leaves is for photosynthesis. Right? Now, when we look at angiosperms, the reproductive organs for angiosperms is its flowers. Remember when we looked at bryophytes, we saw its reproductive organs was with the mosses. And then when we look at pteridophytes, we saw its reproductive organ was on the prothallus. And then when we looked at gymnosperms, we saw that its reproductive organs was when its reproductive organs developed later on. Right? The female gametophyte, the ovule, when all the three disintegrated and the one then survived and it became the female gametophyte, and blah 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 blah. Now, the flowers of angiosperms are the main parts for sexual reproduction in angiosperms. Now, a flower consists, we actually did this in grade 7. Flowers have three main parts. Flowers are divided into three main parts. First part is your perianth. Second part is your androsium. So it's now not antheridia anymore. It is now androsium. So don't get confused there. Oh no, this is right. Androsium. And then the other one is your gynoecium, which makes sense. Gyno, female. Right? Androsium, andro. Anyway, now your perianth, remember perianth you can also have a perigon, right? Your perianth can consist of the corolla and or the calyx, right? Because it doesn't have to consist of both. The androsium is known as your stamen. It's your male part. And then the gynoecium is your pistil, right? The female part. So what happens here is, I don't know if I can draw this thing. So open your textbook to page open your textbook to page 1.27. At the bottom of page 1.27, that is the drawing that I will not be drawing. So let's just talk about it quickly. Now, can we see? So this is also going to be a nice exercise for you to locate. Can we see that there's something known as the anther, which on the left-hand side of the drawing, we see the anther and we see the filament, right? So I'm going to draw it individually. So we see that there's an anther, and then we see that there's a filament coming out of it, right? So at the top here is your anther. At the bottom there is your filament. And then when we're looking at the female side, we're looking at the right hand side there, we see a long structure there. We see a long structure over there. Oh, terrible. We see a long structure. At the top there, even worse. We see a long structure at the top there. We see at the top part here is your stigma. Then this here is your style, which just makes sense because the stylus, pen stylus is thin. And then here we see the outer layer. Now be careful when looking at your drawing. The outer layer is the ovary. The ovary will always be the ovule or the ova. And then inside of it here is the ovule. Right? Now also be careful when you're labeling your drawing. If they ask you to draw it. Yeah, hopefully not. Only the first three here. Only the first three without the ovule until the ovary. So only the first three here represent your pistil. And then these two over here will represent your stamen. Your pistil being your female sex organs, or together we just call it the female sex organ. And then your stamen being your male sex organ. Now what happens here is, with the stamen is going to, contain its sporangium, right? It's going to have male spores. The stamen, so in pollen sacs, similar to gymnosperms, in pollen sacs, we will have pollen grains or male spores, right? So you'll have a pollen sac. The process is sort of similar. You have your pollen grain, This is now the stamen. Right? And then, when we're talking about the pistil, now this stigma, pardon me, this stigma here is sticky. Keep that in mind, that this stigma here is sticky. Right? Now what happens is, when the pollen sac bursts, so when the pollen sac bursts, that pollen will travel that pollen grain will travel to the stigma. That pollen grain will land on the receptive stigma. It attracts it till there. Right? That process here, where this pollen grain goes to the stigma, is known as pollination. The stigma releases a sweet sticky substance to attract insects to it, bees, ants, or anything. Right? That is why it releases a sticky substance. It was going to land on the stigma. What happens now is a similar process happens. Yo, I think I must not draw it. Oh, sure. I'm not going to draw it. You just have to if you just look at the drawing there, I'm just going to draw it more simpler. It's going to land there. That pollen grain over there, it's now going to germinate and it's going to develop a pollen tube leading, it will develop a pollen tube with 2 male gametophytes. And that pollen tube is obviously going to grow down. That would be the pollen tube developing. At the top here, you'd have that germinating pollen grain. Germinating pollen grain, the pollen tube will now grow down. That pollen tube contains 2 male gametes. Now there at the bottom there we see of the female spores. Right? Now one of those female spores, okay, I think I'm going to have to draw it now, because the drawing in the textbook is a bit hectic. Okay, I'm going to enlarge it now. So, if I just draw it, after a very lengthy process, it's finally drawn. So, remember I said that the pollen tube is going to go down with the 2 male gametophytes. That's the pollen tube there, and that's the 2 male gametophytes in it. Right? And then, okay, so this here is now that pollen tube. And these 2 structures here are your 2 male gametes or gametophytes. Right? Your 2 male gametes. Now, what happens is, one of those female spores, so one of these female spores here, they create an embryo sac, which is this outer layer here. This layer here, they create an embryo sac. Right? They create an embryo sac. And inside the embryo sac, there are ovum and there are 2 polar nuclei. Right? There are ovum and there are 2 polar nuclei that are fused together, and that is this here. That is that 2 polar nuclei. That is the two polar nuclei together. So, and then obviously here, this here would be an ovum, one singular ovum. So what happens is the pollen, as I say, the pollen tube will go down the style, right? And it will now go into the ovary. It will then penetrate the, so it will go, so if we just enlarge this, if we enlarge that, if we enlarge, okay, let's just enlarge the style. What happens is the pollen tube grows down the style, it penetrates the ovary, it now enters the ovule, right? And then it will eventually reach the embryo sac. So now it is now in the style, is now in the ovule, and now it's going to the embryo sac. Once it's at the embryo sac, it will release that two male gametes. That two male gametes will be released. The reason I draw it to two different spots is because two different things happen. One male gamete will fuse with an egg cell, with an ovum. One male gamete will fuse with an egg cell. So, this egg cell and that gamete there, that egg cell and that gamete will fuse, right? And they will develop a zygote. And the zygote will then become an embryo. And that embryo will then develop into a seed. Right? But then, the two polar nuclei will fuse together with that male gamete. The two polar nuclei will fuse together with that male gamete over there, and they will develop the covering of the seed, which is also known. It will first become an endosperm. They will develop an endosperm. After they develop an endosperm, they develop something which is known as the tester, and that tester will then be the covering of the seed. So, with a gymnosperm, we would have just had a normal seed like this. Normal seed. Right? A naked seed. However, with angiosperms, you now have a naked seed, but you also have around the naked seed, which is this part here, you have a covering there around it, which is known, a hard covering there, which is known as the tester. So that is the main difference there. Here, in order for this to happen, when we looked at gymnosperms, we saw that it was single fertilization. This is what I mean when I say single fertilization. Right? It was an ova and a sperm that fused together, or a gamete you say. Alright? And then here, it was double fertilization. Because in order for this part to develop, we saw an egg cell fuse with a male gamete. Then, in order to develop the outside part, we saw the two nuclei, right? The polar nuclei, the two polar nuclei fuse with the other male gamete. So can we see here fertilization took place and here fertilization took place, but it's different things. This one developed a zygote, embryo, a seed. This one developed an endospore, an endospore, and then the tester. And that is the biodiversity of plants. So obviously you can go and rewatch the video over and over and over until you understand it. I hope I made it easy to understand. Yeah, so that is your biodiversity of plants without pollination.